So thank you, Liz. Great talk indeed. 4 great talks. So I'd like to invite all the panelists up now for Q&A and discussion. Plus, we're also going to include Linda House from the Cancer Support Community and Louise Perkins from the Melanoma Research Alliance to join us. So to start out with the questioning and discussion, maybe let's start with Linda and Louise, since you haven't had a chance to speak yet. And we really haven't heard a whole lot today so far from the advocate cancer care nonprofit community. So I guess I would ask the two of you if your thoughts on whether there are additional factors related to the design of clinical trials, combination clinical trials, that we haven't discussed yet today. Issues, quality of life, toxicity, cost, access. I know there are a lot of other issues that'll impact as we think about combination therapies.
Sure, I'll go ahead and start. And just to give you an overview, our organization is a 35-year-old organization. We have 170 locations around the world. We have a research and training institute in Philadelphia where we have clinical psychologists and social scientists studying the cancer patient experience. And we have a public policy institute. And I only share that with you, and my background is I'm an oncology nurse by training and practice. But I share that with you because we try to really sort of bring all of these elements together and help shape the way that you all are thinking about clinical trial design, and also shape the way that regulators are thinking about decisions that they have to make for the future, and in particular payment mechanisms and other things. And what I think about as I've been sitting here today listening to the conversation is how really the patient has been missing from the conversation. We've done a really nice job talking about how we're treating tumors and how we're really targeting tumors to shrink tumors, but we really haven't talked about how our patients living with their cancer and living with the treatments that we're giving them. And so some of the work that we've done with our FDA colleagues in the room is to really think about how do we integrate this concept of patient experience outcomes into clinical trial design. And I wanted to really raise that here with you because it's at this stage that we have to collect the data, because decisions are being made downstream that really impact patients without having the benefit of having this type of data. So just a quick example of that is when you look at either the ASCO value frameworks, or when you look at the NCCN evidence blocks, they're making decisions on toxicity, efficacy, cost, based on clinical trial data that doesn't contain information that would really give them the ability to make informed decisions about those elements of care. So, when I sit here and listen to us talking about tumors, thinking about the patients that we're going to end up picking up, and trying to help meet some of these needs, one of the things I'd like to continue to evolve this discussion around is how do we start to integrate these patient experience outcomes into what we're actually measuring. And I just looked up quickly because we have a cancer experience registry where we've got over 13,000 patients who are answering questions about their cancer experience. It's an IRB study, and we publish from that pretty frequently. And I just looked at the last publication that we've done in melanoma and in lung cancer and looked at just the top 5 concerns. We've got a validated distress screening tool that we were looking at. This whole idea of being physically active, this whole idea of fatigue, the ability to eat and maintain nutrition, this worry about future financial toxicity, right? Those are the things that we aren't collecting when we're thinking about toxicities related to the care that we're providing to patients. So I'll just leave it at that for now. I'm sure we'll get into more of that conversation, but I appreciate the opportunity to sort of be here and to help have you think about some of the other important elements as you're thinking about clinical trial design. So thank you.
And my name is Louise Perkins. I'm the Chief Science Officer at the Melanoma Research Alliance, and I'm partly here to represent that not all nonprofits are created with the same orientation, but I absolutely positively agree with everything that Linda just mentioned, that the customer needs to be at the center of the discussions. Why would you build a product without customer input? So that is critically important. But beyond that, I have to say our input as customers into the process derives from the fact that we were founded by a patient, Deborah Black, shortly after her diagnosis with melanoma. This was in 2007. And the time since then, we've focused on funding research, so we've become the largest private nonprofit funder of melanoma research and have invested $101 million in research programs to academic institutions around the world. Beyond the funding, we work with a lot of the folks who are here in the room, the industry partners, the FDA partners, the academic partners, as well as patients to help continue to progress things in melanoma. I wanted to just talk for a minute about the kinds of programs we fund because it comes back to the theme for this meeting of combinations. And we fund early career investigators, we fund established investigators, we fund pilot programs, and a lot of what we fund, actually about 2/3 of the research we fund, is team science. So team science depends on combinations of researchers. And some of those combinations of researchers are akin to some of the things that are taking place in combination immunotherapy approaches right now. They're combinations of convenience, things that are easy to do because it's A plus B, and why not? We don't really have a good reason to select for them otherwise. Some of them are combinations of great ideas that have grown, great people with ideas that have grown up organically and really propel themselves forward. And I think an increasing emphasis, as has been talked about today, some of the data that Adil showed, with a strong hypothesis and then data that supports it to move forward a particular combination can really be applied increasingly more, and we would be very much in favor of that, to use a more rational approach to selecting combination clinical trials, because I'm probably an outlier in the room who thinks that, in fact, there may be too many clinical trials going on right now in the immunotherapy landscape. I think there are some lower hanging fruit that could be trimmed, and that would potentially free up resources and speed things in the system otherwise. But beyond selecting the combinations themselves, I think I wanted to just leave you with some of the other aspects where combination approaches could really have great benefit. For example, treating brain metastases concomitantly with visceral mets. From what the data I've seen, certainly in the melanoma verse, you may be able to treat one, but the other one may not be effectively managed. There was an effort also, hearkening back to what Linda mentioned about getting patients more in the process. There was an effort put forward by ASCO and Friends of Cancer Research to modernize clinical trial eligibility, and I participated, along with probably some other people in the room, in a number of aspects of that. And our particular section was on incorporating patients with brain mets into clinical trials. The notion being, across all of that, why not have the customers, a more representative group of your customers, included in the early clinical trials, so that you don't just know how your drug works in the marathon runners, so to speak, of the clinical trial universe, but actually in a real world population, and not to pollute the clinical trial to the point that it disables it or you may damage an approval, but rather you have that information because you know that mom and pop in middle America aren't going to be seen by a leading center with experience treating people with a lot of comorbidities. So I wanted to raise that as an important issue for us, brain mets in particular, and including real-world patient populations. Developing endpoints, I think, was mentioned, especially endpoints around adjuvant therapy, neoadjuvant therapy. And I think there's room. The FDA has done a great job, in my opinion, of taking calculated, scientifically based risks. And I think there's room for that in the biomarker world, that maybe you're not selecting a biomarker that is going to predict overall survival, but maybe is a calculated risk towards prioritizing. And finally, I'll just wrap up by encouraging you to approach your friendly neighborhood nonprofit, whether it's to get the patient perspective or other perspectives and other connections.
Thank you both. Questions? Well, maybe I'll start out and say, we were talking today about clinical trial design for PD-1, PD-L1 combination therapies, and I'm wondering what is unique about that compared to other combination therapies. So what should we be considering that's different than when we're combining two targeted therapies or a targeted therapy and a chemotherapy when we're dealing here with IO? What are unique considerations?
I mean, the one, I'm just going to take a stab at this, although I'm probably not the right person to answer this, but I think just to pick up on what Louise was saying, I think for a long time we've treated brain mets as an exclusion criteria in clinical trial, but maybe the time is coming now to not do that and just let patients with brain mets get on clinical trials. Because we now know that in the era of checkpoint inhibitors, it probably doesn't make sense to separate those out, and kind of like send them to the leper colony. What we don't get data that way, we don't learn more. And, some of these distinctions, I think what I personally feel is that we need to really think hard about the exclusion criteria and not exclude so many patients. I mean, in our clinics, I think we put 20% of the patients who actually walk in on clinical trial, and I think that is considered better than the national average. But there's a lot of people there who could potentially be put on, who we are just mindlessly excluding because we have A and C, or I can't even remember all the different exclusion criteria we use.
In terms of some of the unique features, let's say in terms of immunotherapy, I would think first in the endpoints. When we talk about, let's say, response rate, overall survival, relapse-free survival, obviously using classical criteria in terms of assessing response, let's say with immunotherapy, RECIST, should we be using immune response criteria or modified RECIST criteria? And these are being used. They're still considered experimental, but understand that because we're still learning, from immunotherapy and combinations, but probably can be used in combination with classic criteria in terms of the decision making. And I think this is being done anyway. But these are some of the features. Other things are the biomarkers. We're talking targeted therapy. We have, let's say, BRAF mutation. That's a predictive biomarker. In combination with MEK inhibitors, 70% will respond. Biomarkers in the immunotherapy world are more dynamic. The microenvironment is more dynamic. When we introduce other agents, things change. Are these truly predictive biomarkers or more of in terms of, let's say, enrichment strategies for response to immunotherapy or not? Can we design? Can we believe an adaptive design which says, based on this biomarker or this signature biomarkers, we need to exclude those patients from this combination versus that combination? That's a challenge, I think, something that we're still learning. Eligibility criteria, obviously another thing.
Yeah. Thank you. Yes, sir.
John Gordonier, retired from the National Center for Health Statistics, but my issue is about science in general, and particularly cross-silo communication. Obviously, you have very complex problems which you're dealing with in very rigorous and determined ways, and that's very good. The same thing is true in most every other field of science as well. And as a result, there are different organizations that are specifically set up to try to deal with data science and research issues across the silos. My question is for you and for the National Cancer Policy Forum generally, to what extent are you taking advantage of organizations such as the National Academy of Sciences, Board on Research Data and Information, for one, and the Global Research Data Analysis for another, or any other similar things? Or have you looked at that and found it not to be a productive area of examination?
Any volunteers? I mean, we certainly have the presidents of two major organizations that are involved in data sharing and interdisciplinary research, and Liz and Lisa.
So I guess for SITC's part, we have not worked with the organizations you specified, but I think one of the points of a professional membership society in this field is to create task forces and to bring people who are members of the society from pharma and academia, from government and regulatory together on task forces to cross-pollinate and fertilize those ideas.
Yeah, and I think your point is extremely important, and the American Association for Cancer Research is now focusing on what we're calling convergent science, but computational biologists from all different realms, engineers, we're trying to bring them in, and we actually have a convergence symposia in September, and that's probably going to be a regular annual meeting. And I think that it's going to be big data heavy.
Dan Chen, Genentech again. Question around dose, schedule, duration of therapy. So we recognize the immune system as being a highly dynamic biologic organ, essentially, within our bodies. We know that single agent already faces lots of questions as it relates to how much you give, when you give it, and for how long. I think some of you already touched upon it. As we get into combination therapies, whether it's 2 or 3 immunologic agents or agents that affect the immune system, you start to get into a whole different type of a problem. And yet, at the same time, it can be critically important to the outcome of a combination trial. So what do? How do we face this problem?
So I think that's why we do phase 1 studies. And
we could literally spend 3 to 5 years doing these phase 1s if we start to test these critically important questions. And I think that's the crux of what we have to face for combination immunotherapy.
I think, what we're doing is we're going to have our best guess, in phase 1 studies moving forward. In terms of the dose and potentially duration, I think the initial strategies of treating for 2 years were right. And now is the time to go back and say, and I know there are discussions within ECOG about doing a study which is histology agnostic, treating with anti-PD-1 for 6 months versus 2 years, and see, let's say melanoma, lung, etc. Do we really have to treat for 2 years? But in phase 1, I agree these are challenging questions that we're going to have our best guess, in my opinion.
I worry about that too. I think it's a really important question. And one of the areas that's absent from our field, I think, is pharmacodynamic studies. And I really think we need to be thinking more about that in this setting. And I think that would be an area to try to maybe even have a conference around.
What about the patient perspective on how long, do you risk stopping too soon, or do you risk going too long with these therapies?
I think that's an individual question, truthfully, because then unfortunately it's the patient who's sort of thinking about what they're willing to tolerate and for how long are they willing to tolerate it. And one of the things that we're learning, we're doing thanks to a generous grant from BMS. We have a clinical trial ongoing in 5 sites where we are trying to really understand the patient experience as they're going through their therapies. So we are we've taken the best of what CSC has to offer around education and materials. We have a nurse on the helpline, anything that really sort of supports the patient in that journey so that we can help them through. We've created an online digital symptom tracker. We've created our distress screening tool baked into that, and then educational materials. So the idea is can we track what their physical symptoms are at particular points of time throughout their a 6-month period of time? Do we correlate those with any sort of major events? Can we correlate those with any sort of spikes in distress over a 25-item tool? So it'll be the first data set of its kind that's really collecting this type of information for patients on IOs, and we hope to have some preliminary data, hopefully at ASCO-SITC next year. But I think that it will help us start to get this little insight into, okay, where are those hot points? Where might too much be too much? And we just don't have that information today.
Thanks. Hi, Dave Ram from Yale. I'm particularly aiming this question at Dr. Butterfield. You talked about the SITC actions to monitor and improve the quality of biospecimens. Is there going to be some kind of best practices out of this group, or could there be in terms of when we're asked for biospecimens, we're, send a block, but we can't send a block. We're obliged to keep the block as pathologists, so we send sections. Then the sections get lost. And then I talked to some of my farmer colleagues, and they say, oh, no, we can't do that trial because it wasn't consented for that experiment. They were only consented for the experiments that we were integrated into the trial. And is there any way that, perhaps led by SITC or maybe AACR, you could have a system where there was sort of a best practices that perhaps pharma or anyone doing a clinical trial could follow? And can you comment on what your plans are related to that?
So one of the things we've been working towards is having SOPs on the website as a shared resource so that people can access, the publications that people have agreed upon are great, valid ways to process, blood and tumor for immunologic questions. That's the easy part. The consent part and what was approved. This is, this is a battle I fight myself fairly regularly with my clinical PI's help to be able to ask new questions that even when we can get them funded, that wasn't in the consent, and the hurdles. You figure you'd just rather retire now than climb those administrative hurdles to ask that really important scientific question to ask today. So that I look for consensus amongst all of us on how to handle that.
One suggestion is, in each institution is to have a consent in place, kind of broad consent that's offered to the patient who are willing to participate in doing, studies using the biospecimens collected on any study, basically. It is, there's something similar in Pittsburgh, for example.
Roy Herbst from Yale. I have a two-part question. The first is, given what we've heard today about trial designs, diagnostics, some of the drugs, tomorrow we'll hear about data, are we ready to do an adaptive trial where patients would come in and in a CLIA environment have their tumors profiled and then assign them to different arms of the trial? So that's question one. And then who would do it? So from a policy issue, would it have to be a public-private partnership? Would a company be driving it? Would one of the societies drive it? Are we ready for that yet, or are there barriers?
So I think we heard that Bristol Myers Squibb is already getting ready to do one, and they needed, because I've been advising them on it, they're doing 8 different checkpoints, I believe, and they needed to develop CLIA-level assays before they could start. So that took them probably almost 2 years.
Any other questions? Any other comments from the panel? Issues we haven't discussed? We have a few minutes.
I was going to try to take a stab at the question that Dan asked earlier on. I think one of the problems for me has been just the complexity of how these things work. Like IPI, it's been around since 2000, since for a while now, but we are still learning things. Does it deplete Tregs? Is it working through some other mechanism? We don't fully understand that. But I do think, in general, the pendulum is, in my mind anyway, it's swinging towards shorter courses of treatment. And especially once somebody has achieved a complete response, I feel like there isn't that much more to be gained by continuing treatment. But then I see the data for lung cancer, where essentially PD-1 behaves a lot like a chemotherapy drug. It's not like this is just my secondhand look at the data. It doesn't seem like patients are cured, for lack of a better word. It seems like you're kicking the can down the road to some extent. But so I think some of what, in terms of trying to figure out the right duration of treatment and schedule and stuff like that, and that HODI trial, to me, was a pretty eye-opening kind of thing, because just flipping the order of IPI and NIVO, for that to make such a big difference, kind of suggests that we don't really understand. I think it's hard to square that with IPI priming, because if it was priming, why shouldn't it? It should have worked in that setting, right, in the pre-treatment. So, not a real answer, but just a rambling kind of,
And I just wanted to build on that, because I think one thing we haven't talked about is what will the advent of adjuvant checkpoint immunotherapy have? What will be the influence of that on patients? Because we know that these treatments can permanently rewire diabetes, thyroiditis, vitiligo. So there are probably other things that are being permanently affected in individuals that we're not sensing yet. So as people are being treated in the adjuvant, or indeed in the future in the neoadjuvant setting, what does that mean for either them as individuals or for their disease? And does it mean that we need to be considering the tissue that we're analyzing in a different light compared to, say, the TCGA tissue?
Lisa, did you?
And I guess I want to circle back to Dr. Rim's question. One of the other things that, as I think about what Dr. Tarhini just said, some institutional IRBs have allowed, more open use of specimens. And one of the things we did in the first recommendations paper from 2011 is in Pittsburgh in the melanoma program, we routinely draw 160 cc's of blood every time the patient comes in, all for research. And Nora Desis had done a test in stage 4 breast cancer patients where 250 cc's was approved, and she could routinely get 200 cc's with no adverse effects for the patients, their hematocrit, anything. And so we tried to also put those on the web so that other IRBs, people could cite those as perfectly reasonable things to have approved. And so that might be, another way that the societies can pool, success stories from individual institutions to make this more broadly available.
Okay, so I'd like to thank the speakers and panelists for a great discussion, and I believe we have a 10-minute break before the final session of the day.